This is part 3 of Chapter 5's lecture on biomolecules, and we left off talking about carbohydrates or sugars. And so the monomer, again, of a carbohydrate is a monosaccharide. And now as we get beyond disaccharides, which would have two monomers, we get into polysaccharides, which can have hundreds to thousands of monosaccharides joined together by glycosidic linkages. So that's the bond between the monomers of carbohydrates. And these polymers of sugar are excellent storage molecules for energy because they don't take a lot of energy to build, although, of course, we know the process of photosynthesis is a lengthy one. But they're also easy to break down and release their energy. So their function is broken into two main categories. Either they act as an energy storage molecule or they function as a structural molecule. And then those two categories break down into two other categories. So there are two types of energy storage molecules. There's starch, which we find in plants, and glycogen, which is found in animals, and particularly in the liver and muscles of animals. So those are the two types of energy storage polysaccharides. And then in terms of structure, we have both cellulose and chitin. And cellulose is what keeps a plant upright. It's what gives it its structure. And chitin is found in arthropods and fungi, arthropods being insects. And what the chitin provides for these organisms is a hard coating to protect it. So, when you pick up an insect and it has that hard shell, that's the chitin. Or it's the hardened outer casing of a fungus that help protect it. So the energy storage molecules of starch and glycogen actually can take different structural forms. There's either a linear or a branched form of a polysaccharide. And starch is more of the linear form, and glycogen is more of the branched form. So again, animals store glucose in a polysaccharide called glycogen, and they essentially have a day's supply of glycogen in their liver and muscles. So that's enough to break down and supply the body for a day's worth of energy. And that energy is obtained by hydrolyzing the polysaccharide to release the glucose to meet the body's demand for sugar. So glycogen has many branches, many ends, and it's actually what the term glycogen means literally is many branches. And what's nice about it is that an enzyme can digest it at these multiple branches at the same time, which is what allows glycogen to be a fast release molecule. It's because the enzyme can work at the different ends of the branches. So, and then if you look at starch, an enzyme is only going to be able to work at the two ends to break it apart. And so starch is a much slower released polysaccharide. So it's much more difficult to digest, in essence, because if you remember, to digest means to break apart. So molecular structure determines its function. And what we find with carbohydrates, just as we find with many other of the biomolecules, there can be isomers. So in terms of glucose, we have the alpha and the beta form of glucose. And it is simply going to be a structural isomer where the hydroxyls are reversed in this one spot, at the one carbon spot. But these two ring forms differ in terms of giving a different function to these two types of glucose. Now, the significance of these two different forms of glucose are really interesting because starch, as we were talking about the difference between starch and, for example, cellulose, starch is an energy storage and cellulose is a structural polysaccharide. And starch is actually made up of alpha glucose monomers, and cellulose is made up of beta glucose monomers, which makes every other glucose monomer upside down with respect to its neighbor. So that ends up actually establishing different glycosidic linkages in starch as opposed to cellulose. So that in turn gives the two molecules a distinct three-dimensional space or shape. So polymers built with alpha glucose, which are the starches, take a helical structure, and polymers built with the beta glucose form a straight structure, which is interesting because if again you think of the different functions of these two polymers or polysaccharides, it actually makes sense for the beta glucose to form a straight structure if it's a structural polysaccharide. And if it's actually just being used for food storage or energy storage, to have it be helical will actually allow you to cram more of the energy storage into a smaller space. What's also important in this distinction is that enzymes that digest starch work at hydrolyzing the alpha linkages, but those same enzymes cannot hydrolyze the beta linkages in cellulose. So that's why when humans, for example, eat cellulose, we actually cannot digest it. It actually, when you hear about, eat your roughage, make sure you have roughage in your diet, what they're talking about is cellulose, because cellulose is something that's going to pass through the entire digestive tract without ever being digested. It's actually never going to be broken apart because human enzymes in the digestive tract only break apart the alpha linkages found in starches. The only organisms that can actually break apart the beta linkages or the beta form of the glucose are those that have some bacteria or microbes that are able to digest those type of linkages. So they're able to digest cellulose. So cows, for example, will have these microbes in their digestive system, or things like termites, that eat wood, which is made up of cellulose, are able to digest cellulose because they have these particular microbes that actually have enzymes that will digest the cellulose. So that's actually what this is talking about here. So you can pause this and just study how these look. And this little gray cloud is actually trying to show the different enzymes that would actually work in terms of digesting each of those and how they're different. Okay, so we're going to move on now to nucleic acids. And the main role of nucleic acids, as I'm sure you know, is to store information and to pass it on and to allow it to be expressed. So they contain the genetic material. As you know, they are what genes are made up of. Genes are the blueprint for building proteins. So the central dogma, again, is DNA is made into RNA, which then is made or used to make proteins. So there's a transfer of information according to the central dogma from DNA to protein. And DNA also, or nucleic acids, provide the blueprint for new cells. And they're also what pass on from generation to generation to offspring the genetic information. So the monomer of a nucleic acid or of DNA is a nucleotide. And a nucleotide is made up of three parts. There's the nitrogen base or nitrogenous base, which is a carbon-nitrogen ring. There's a pentose sugar, which is a 5-carbon sugar. It's either ribose in RNA or deoxyribose in DNA. And then the third part is a phosphate group. So if you look at the molecule down here, you can see these three parts, and they're color-coded so you can see what they look like in a DNA polymer. And the question we have here, looking at this molecule of this monomer, this nucleotide, would you say a nucleic acid is a charged molecule? And hopefully you'll remember that yes, it is. Nucleic acids are negatively charged, and that has everything to do with the phosphate group. So if you remember again, a phosphate group with these 4 electronegative oxygens is a very highly electronegative functional group. And so that negative functional group makes the entire DNA molecule negative. And that's important because it keeps the DNA inside the nuclear membrane. Because if you remember, again, to regular biology, we'll do cell biology in our next unit, but hopefully you can remember back to early biology that things that have a charge, like ionic compounds or large molecules, cannot pass through cell membranes. And so since DNA and RNA are negatively charged, they're not able to pass through the nuclear membrane into the cytoplasm. And that's really important. It keeps it where it's supposed to be. But of course, you also know that RNA has to get out of the nucleus in the form of messenger RNA or ribosomal RNA. Those things have to get into the cytoplasm. So there are special proteins in the nucleus that are actually responsible for transporting the RNA across the nuclear envelope. Another important just side note about this negative charge is it's the reason that we're able to do gel electrophoresis, or the separation out of DNA, and actually everything we know about forensics and DNA at this point is due to this ability to use the negative charge in DNA to separate out DNA and gel electrophoresis. And when we study biotechnology later in the year, we'll actually do some of this, so we'll learn a lot more about it then. So back to the monomer of DNA, there are 2 types of nucleotides. They have, of course, you know that there are 4 different nitrogen bases. There's adenine, guanine, cytosine, and thymine in DNA, and then cytosine and uracil in RNA. But these are actually broken into 2 different groups of nucleotides. And those 2 groups are the purines and the pyrimidines. And you should know the difference between them, which are which. So the purines are simply a double ring nitrogenous base. So they're larger bases. And they are specifically adenine and guanine are the purines. So you can see that clearly in the picture here, that they're a 2-ring structure. And the pyrimidines are just a single ring structure. And they're made up of cytosine, thymine, and uracil.